Welcome to Dream Lab Knowledge, where tonight we embark on a journey through time itself, traveling backward through billions of years to witness one of the most profound mysteries in all of science: the origin of DNA, the elegant molecular code that defines every living thing on Earth. We exist in a world where DNA is everywhere, coiled inside the nucleus of nearly every cell in your body, dictating the color of your eyes, the rhythm of your heartbeat, the shape of your hands. It stores the instructions for building proteins, for repairing damage, for creating new life. DNA is so fundamental to our understanding of biology that we sometimes forget how extraordinary it is that this molecule exists at all. How did this double helix, this spiral staircase of genetic information, first come into being? Where did the code come from? These questions take us back to a time before humans, before animals, before plants, before even the simplest bacteria. They take us back to an Earth that would be unrecognizable to us today, a planet still cooling from its violent birth, its surface pockmarked with craters from asteroid impacts, its atmosphere toxic, its oceans potentially boiling. Somewhere in this hostile environment, through processes we are only beginning to understand, simple chemicals combined and recombined until they crossed a threshold that separates the inanimate from the living. And somewhere along that journey, DNA emerged. Tonight, as you settle into sleep, let your mind wander through deep time. Let yourself imagine an Earth without grass, without trees, without insects or birds or any of the million forms of life we take for granted. Picture instead a world of rock and water and churning chemistry, where the boundary between geology and biology had not yet been drawn. This is where our story begins. The puzzle of life's complexity. To understand the origin of DNA, we first need to appreciate just how astonishingly complex this molecule is. DNA is not a single substance, but a polymer, a long chain made up of smaller units called nucleotides. Each nucleotide consists of 3 parts: a sugar molecule called deoxyribose, a phosphate group, and one of 4 nitrogen-containing bases: adenine, thymine, guanine, or cytosine. The sequence of these bases along the DNA strand encodes genetic information in much the same way that the sequence of letters in a sentence encodes meaning. But DNA doesn't work alone. To function, it requires an entire molecular ecosystem. It needs enzymes to unzip the double helix, to read the genetic code, to make copies of itself. It needs RNA to carry messages and to help build proteins. It needs proteins to do almost everything, to catalyze reactions, to provide structure, to regulate gene expression. This creates what scientists call the central dogma of molecular biology. DNA makes RNA, which makes proteins, which in turn help make more DNA. Each component depends on the others. DNA needs proteins to replicate, but proteins need DNA to provide the instructions for their construction. This interdependence presents a profound puzzle. How could such a system have originated? It's like encountering a fully functioning car in the middle of an empty desert and being asked to explain how it got there, but with the additional constraint that you can't invoke a factory or a designer. The car must have assembled itself from raw materials. Where would you even begin? For much of the 20th century, this puzzle seemed nearly insurmountable. The discovery of DNA's structure by Watson and Crick in 1953 was a triumph of molecular biology, revealing how genetic information could be stored and copied. But it made the origin of life seem even more mysterious. How could something so intricate, so specific, so apparently designed, arise from random chemical reactions? The RNA World Hypothesis. The key insight that began to unlock this mystery came from an unexpected direction. In the early 1980s, researchers made a discovery that would revolutionize our understanding of early life. They found that RNA, DNA's chemical cousin, could do something that had previously been thought impossible for a nucleic acid. It could act as an enzyme, catalyzing chemical reactions. This discovery earned its discoverers, Thomas Keck and Sidney Altman, the Nobel Prize in Chemistry in 1989. They had found molecules called ribozymes, RNA sequences that could cut, splice, and modify other RNA molecules without the help of proteins. The ribosome, the molecular machine that builds proteins in every cell, turned out to have an RNA molecule at its catalytic core. Here was RNA doing the job of a protein, bridging the chicken and egg paradox that had plagued origin of life research. The discovery of catalytic RNA led to a radical proposal. What if, before the modern world of DNA and proteins, there was an earlier, simpler stage of life based entirely on RNA? This idea, championed by molecular biologist Walter Gilbert, who coined the term in 1986, became known as the RNA World Hypothesis. According to this scenario, RNA once performed both roles that are now divided between DNA and proteins. It stored genetic information like DNA does, and it catalyzed chemical reactions like proteins do. The RNA World Hypothesis is elegant because it solves the interdependence problem. You don't need to explain how DNA and proteins arose simultaneously if RNA came first and did both jobs. Only later, according to this scenario, did the more sophisticated division of labor evolve, with DNA taking over information storage and proteins taking over catalysis. RNA would have remained as a molecular fossil, a bridge between the ancient past and the present, still playing crucial roles in translation and gene regulation. Evidence for the RNA world comes from multiple sources. RNA is less stable than DNA, which at first seems like a disadvantage. But this very instability would have been useful in an early evolving system where flexibility and rapid turnover were important. RNA uses ribose as its sugar backbone, while DNA uses deoxyribose, ribose minus one oxygen atom. This suggests that DNA is a chemical modification of RNA, not the other way around. From a biosynthetic perspective, DNA appears to be derived from RNA, a stripped-down, stabilized version optimized for long-term information storage. Modern cells still show evidence of RNA's ancient prominence. The process of making DNA nucleotides involves first making RNA nucleotides and then chemically modifying them. Enzymes called ribonucleotide reductases remove the oxygen atom from the ribose sugar to create deoxyribose. This metabolic pathway makes sense if RNA came first. The chemistry of life would have been built around RNA, and DNA synthesis would have been retrofitted into an existing RNA-based system. Moreover, many of the cofactors and coenzymes that drive metabolism today are built around RNA-like structures. ATP, the universal energy currency of cells, is essentially an RNA nucleotide. So are NAD and FAD crucial molecules involved in energy metabolism. It's as if the entire metabolic machinery of the cell was originally constructed from RNA components, and these components were never fully replaced, even as evolution moved beyond the RNA world. Before the RNA world. But recognizing that RNA might have preceded DNA only pushes the mystery back one step. Where did RNA come from? How did it emerge from prebiotic chemistry? This question has occupied researchers for decades and remains one of the most challenging problems in origin of life research. RNA, for all its advantages over DNA as a primordial molecule, is still dauntingly complex. Each nucleotide is a three-part structure that must be assembled in the correct sequence. The sugar, the base, and the phosphate must come together in a specific geometry. Then these nucleotides must link together in long chains, with each new bond requiring energy and forming water as a byproduct. a reaction that doesn't happen spontaneously in water-rich environments. In the 1950s, a landmark experiment offered hope that life's building blocks could form through simple chemistry. Stanley Miller, then a graduate student working under Nobel laureate Harold Urey at the University of Chicago, set up an apparatus designed to simulate the atmosphere of the early Earth. He filled a glass chamber with methane, ammonia, hydrogen gas, and water vapor, gases thought to have been present on the primitive Earth. Then he sent electric sparks crackling through this mixture, simulating lightning. When Miller analyzed the contents of his apparatus after running the experiment for a week, he found something remarkable. Amino acids, the building blocks of proteins, had formed spontaneously. The Miller-Urey experiment, as it came to be known, seemed to show that the organic molecules necessary for life could arise naturally from simple starting materials and energy input. It was a sensation, making headlines around the world and inspiring generations of origin of life researchers. However, subsequent research revealed that making nucleotides, the building blocks of RNA and DNA, is considerably harder than making amino acids. The Miller-Urey experiment didn't produce nucleotides. Other experimental setups using different gas mixtures and energy sources have managed to create some of the components of nucleotides, but assembling these components into complete nucleotides has proven extraordinarily difficult. The challenge isn't just making the parts, it's making them come together in the right way. The sugar component, ribose, can exist in multiple forms, and only one specific form works in RNA. The bases have to attach to the right position on the sugar. The phosphate has to link sugars together in a specific way. Each step has its own chemical obstacles. For decades, these difficulties led some researchers to wonder whether RNA itself might have been preceded by something even simpler, a genetic molecule that we haven't discovered yet, something that later gave way to RNA. But in recent years, experimental chemists have made remarkable progress. In a 2019 paper published in the journal Nature, researchers demonstrated that RNA and DNA nucleotides could potentially form simultaneously from the same prebiotic soup, rather than RNA necessarily preceding DNA by a vast stretch of time. Their work showed that under certain conditions, nucleotides could self-assemble from simpler molecules in ways that had previously seemed impossible. Researchers have found that mineral surfaces can play a crucial role in organizing and concentrating organic molecules. Clay minerals, for instance, can help arrange nucleotides in sequences, protecting them from degradation and even catalyzing the formation of longer chains. The discovery that borosilicate minerals can dramatically increase the yield and diversity of organic molecules in Miller-Urey type experiments suggests that the right mineral environments could have been molecular nurseries for early genetic materials. Some scientists have proposed that life might not have originated on Earth at all, or at least that some of life's building blocks came from space. Meteorites have been found containing amino acids and even nucleobases. The Murchison meteorite, which fell in Australia in 1969, contained more than 90 different amino acids, including 19 found in Earth life. Comets and asteroids could have delivered organic molecules to the early Earth, seeding the planet with chemistry that might have been difficult to generate on Earth's surface. One intriguing proposal comes from astrobiologist Stephen Benner, who suggests that Mars, rather than Earth, might have been a better environment for the initial formation of RNA molecules. Mars had abundant boron and molybdenum in oxidized forms and a drier environment than early Earth, which might have made it easier to form certain key bonds. If this idea is correct, the chemistry that led to life might have originated on Mars and then traveled to Earth on meteorites blasted off the Martian surface by asteroid impacts, a scenario called panspermia. It's a reminder that the origin of life might not have been a purely terrestrial affair. The transition from RNA to DNA, assuming an RNA world did exist, the next question is, how did DNA emerge from it? What drove the transition from an RNA-based system to one based on DNA? And why did this transition happen at all? The answer seems to lie in DNA's superior properties as an information storage molecule. DNA is more stable than RNA, primarily because it lacks the reactive hydroxyl group on the second carbon of its sugar backbone. This hydroxyl group makes RNA prone to self-cleavage, particularly in the presence of metal ions and at higher temperatures. RNA can essentially attack itself, breaking apart its own backbone. DNA, with its deoxyribose sugar, doesn't have this problem. This chemical difference might seem small, just one oxygen atom, but it has profound consequences. It means that DNA can form much longer, more stable molecules than RNA. A DNA genome can be millions or even billions of base pairs long, storing vast amounts of genetic information without breaking apart. RNA molecules, by contrast, need to be protected and are generally shorter. For a complex organism that needs to store detailed instructions for thousands of proteins, DNA's stability offers an enormous advantage. DNA also uses thymine instead of uracil as one of its 4 bases. This seemingly minor substitution actually provides another layer of stability and accuracy. Cytosine, one of the other bases, has a tendency to spontaneously convert to uracil through a process called deamination. In RNA, where uracil is a normal base, this creates a problem. How can you tell whether a uracil is supposed to be there or whether it's a damaged cytosine? In DNA, any uracil that appears is clearly a mistake and can be recognized and repaired by specialized enzymes. The presence of thymine instead of uracil gives DNA a built-in error detection system. But if DNA is so superior, why did evolution bother with RNA first? The answer is that making DNA requires sophisticated enzymatic machinery. The conversion of ribonucleotides to deoxyribonucleotides is catalyzed by enzymes called ribonucleotide reductases, which use complex radical chemistry to remove the crucial oxygen atom. This chemistry is so reactive that it would likely destroy RNA molecules if it were performed by an RNA enzyme. The process requires stable proteins. This creates a logical sequence. RNA had to come first because it could be made more easily from prebiotic chemistry. RNA could also, through ribozymes, perform enough catalytic functions to get primitive life started. But as evolution proceeded and protein enzymes became more sophisticated, it became possible to make DNA. Once DNA appeared, its superior stability for information storage meant that organisms with DNA genomes had an advantage over those with only RNA genomes. The DNA-based organisms could maintain larger genomes with less degradation, allowing them to encode more complex traits and ultimately to outcompete their RNA-based predecessors. Evidence for this transition can be found in the DNA replication machinery itself. Intriguingly, the enzymes that replicate DNA in bacteria are completely different from the enzymes that replicate DNA in archaea and eukaryotes. They're not just different versions of the same enzyme, they're unrelated protein families that evolved independently. This has led some researchers to propose that DNA was actually invented twice, once in the lineage leading to bacteria and once in the lineage leading to archaea and eukaryotes. This radical idea, championed by researchers like Patrick Forterre, suggests that the last universal common ancestor, LUCA, the organism from which all modern life descended, might have had an RNA genome or a mixed RNA-DNA genome. Only after Luca's descendants split into separate lineages did those lineages independently evolve DNA as their primary genetic material. The fact that they ended up with different DNA replication machinery supports this scenario. Alternatively, Luca might have had DNA, but the DNA replication machinery was subsequently replaced in different lineages through lateral gene transfer, the swapping of genes between distantly related organisms. Viruses might have played a role in this process. Fortere has proposed that DNA was originally invented by viruses as a way to protect their genetic material from attack by cellular defense systems, and that cells acquired DNA by being infected with these viruses. According to this viral origin of DNA hypothesis, the reason different domains of life use different DNA replication machinery is that they were infected by different types of DNA viruses. Whichever scenario is correct, whether DNA was invented once or twice by cells or by viruses, the key point is that DNA represents an evolutionary innovation. It wasn't the primordial genetic material. It came later, after a complex infrastructure of RNA and proteins had already been established. DNA is, in a sense, a refined product, a specialized molecule optimized for a specific task through billions of years of evolution. The chemistry of early Earth. To truly understand the origin of DNA, we need to consider the environment where all this chemistry took place. What was Earth like when life was beginning? Where might the reactions that led to RNA and eventually to DNA have occurred? 4.5 billion years ago, Earth was a very different world. The planet had recently formed from the disk of dust and gas surrounding the young sun, accreting from countless collisions of smaller bodies. The final major impact, which likely created the moon, would have left Earth's surface molten. As the planet gradually cooled, a crust formed, and water vapor in the atmosphere condensed to create the first oceans. But this early Earth was nothing like the blue marble we know today. The atmosphere contained almost no oxygen. Instead, it was likely dominated by nitrogen, carbon dioxide, water vapor, and possibly gases like methane and ammonia, depending on how reducing or oxidizing the early environment was. The lack of oxygen meant there was no ozone layer to shield the surface from ultraviolet radiation from the sun. UV light, which can break chemical bonds, would have been both a hazard to fragile organic molecules and a potential energy source for driving chemical reactions. The planet was volcanically active, with frequent eruptions releasing gases and heat. The surface was heavily cratered from ongoing asteroid and comet impacts, the tail end of a period called the Late Heavy Bombardment. Some of these impacts were violent enough to vaporize parts of the ocean and sterilize large regions of the planet's surface. Yet between these catastrophic events, there might have been quieter periods when complex chemistry could develop. Into this tumultuous environment, we must place the origin of life. The question is, where specifically? What type of location would have provided the right combination of energy, raw materials, and protection from destruction? One popular hypothesis points to hydrothermal vents deep on the ocean floor. These vents, discovered in 1977, are places where seawater seeps down through cracks in the seafloor, gets heated by proximity to magma chambers or by chemical reactions with rocks, and then shoots back up as superheated, mineral-rich fluid. Two types of vents exist: black smokers, which emit fluids at temperatures up to 350 degrees Celsius, and alkaline vents like those at the Lost City vent field, which emit cooler fluids around 50 to 90 degrees Celsius at high pH. Alkaline hydrothermal vents have become a favorite candidate for life's birthplace among many researchers. These vents create natural chemical gradients with alkaline hydrogen-rich fluid from the vent meeting acidic carbon dioxide-rich seawater. The mineral structures of the vents, full of tiny pores and chambers, could act like a natural laboratory, concentrating organic molecules and providing surfaces for catalysis. The temperature gradients could drive the synthesis of complex molecules. Most intriguingly, the pH gradient across the vent walls creates a natural proton gradient, similar to what modern cells use to generate energy. Researchers like Nick Lane and William Martin have argued that these natural proton gradients could have been harnessed by early life forms even before they developed the sophisticated molecular machinery for generating their own gradients. The vent environment would provide not just raw materials and energy, but also a kind of proto-metabolism, geochemistry that could transition smoothly into biochemistry. The chemistry occurring at these vents today, where hydrogen reacts with carbon dioxide to form simple organic molecules like formate, bears striking resemblance to some of the most ancient metabolic pathways still used by modern microbes. Recent experimental work has supported the hydrothermal vent hypothesis. Researchers have succeeded in creating protocells, simple membrane-bounded vesicles that could represent precursors to true cells, under conditions mimicking alkaline hydrothermal vents. Earlier attempts to create protocells in such environments had failed, but these experiments used more realistic mixtures of different types of lipid molecules and showed that heat and alkalinity actually help these molecules self-assemble into membrane structures. The same harsh conditions that researchers once thought would prevent life's origin might actually have been necessary for it. Yet the hydrothermal vent hypothesis has its critics. Some chemists argue that the dilution problem in the open ocean is insurmountable, that organic molecules would be too dispersed in seawater to undergo the concentrated reactions necessary for forming polymers like RNA. The high salt concentration in seawater can also interfere with certain chemical reactions, and while vents provide energy and raw materials, creating the specific sequences of nucleotides needed for functional RNA is a very different challenge from simply making organic molecules. An alternative hypothesis places life's origin in shallow pools or hot springs on land, rather than in the deep ocean. Darwin himself once speculated about a warm little pond where the right chemicals might accumulate and react. The advantage of land-based environments is that wet-dry cycles driven by evaporation and rainfall can concentrate molecules and drive polymerization reactions. When water evaporates, organic molecules become concentrated, and certain reactions that are unfavorable in solution can proceed. When water returns, it can dissolve and redistribute the products. Volcanic hot springs on land might have provided the right combination of heat, minerals, and wet-dry cycling. These environments would have access to ultraviolet light, which could drive certain photochemical reactions, though UV would also damage delicate molecules. Recent experiments simulating Hadean hot spring environments have shown that cycles of heating and cooling, combined with pH changes from atmospheric carbon dioxide dissolving in water, can promote the assembly and replication of RNA molecules. The truth is, we don't yet know which environment was the actual birthplace of life. It's possible that different environments contributed different pieces of the puzzle, that some prebiotic chemistry occurred in one setting and other chemistry in another, with the products eventually coming together. It's even possible that life originated multiple times in different settings, with only one lineage surviving to become the ancestor of all modern organisms. What we can say is that early Earth offered a variety of environments where complex chemistry could occur. Hydrothermal systems, whether on the seafloor or on land, provided energy and mineral catalysts. Tidal pools and beaches offered wet-dry cycles and mineral surfaces. The atmosphere provided reactive gases and energy from UV light and lightning. Asteroids and comets delivered organic molecules from space. In this rich and varied chemical landscape, the building blocks of life assembled themselves. The road to the genetic code. The emergence of DNA was not just about creating a stable molecule for storing information. It was also about establishing the genetic code, the mapping between DNA sequences and amino acid sequences in proteins. This code is nearly universal across all life on Earth, using the same set of 20 amino acids and the same correspondence between three-letter sequences of DNA bases, codons, and specific amino acids. How did this code come to be? Why these 20 amino acids and not others? Why is the code structured the way it is, with some amino acids encoded by multiple codons, while others use only one or two? These questions touch on some of the deepest mysteries of biology. The genetic code appears to be a frozen accident, a convention that was established very early in life's history and then became locked in place because changing it would be catastrophic. If you altered the meaning of even one codon, you would change the amino acid sequence of virtually every protein in the cell, almost certainly with fatal results. Once the code was established, evolution could tinker with almost everything else. But the code itself became nearly unchangeable. Yet the code is not entirely arbitrary. There are patterns suggesting that it evolved to minimize the damage from mutations. Similar codons often code for chemically similar amino acids, so that a mutation changing one base in a codon is more likely to result in a similar amino acid being substituted, reducing the functional impact of the error. This error-minimizing structure suggests that the code underwent some optimization early in evolution, shaped by selection to be robust against mutations. The genetic code likely emerged gradually, perhaps starting with a much simpler code that used fewer amino acids. Early proteins might have been made from just a handful of the simplest amino acids, the ones most readily available from prebiotic chemistry. As metabolic pathways evolved and cells gained the ability to synthesize more complex amino acids, these were incorporated into the code. The code expanded to include all 20 standard amino acids, but the basic framework was established so early that it became universal. Transfer RNAs, the adapter molecules that match codons to amino acids, might hold clues to the code's origin. Each tRNA has an anticodon sequence that recognizes a specific codon, and it carries the corresponding amino acid. The structure of tRNAs is ancient, built around a core of RNA that may be a molecular fossil from the RNA world. The fact that tRNAs are RNA molecules, not proteins, supports the idea that the genetic code was established during a time when RNA, not protein, was the dominant functional molecule. Some researchers have proposed that the earliest genetic code involved RNA enzymes directly recognizing and binding to amino acids based on chemical affinity. Certain amino acids have a natural affinity for certain RNA sequences. These chemical preferences might have seeded the genetic code, providing an initial imperfect mapping between RNA sequences and amino acids. Over time, this direct recognition system would have been replaced by the more complex system involving tRNAs and amino-acyl tRNA synthetases, the enzymes that attach amino acids to tRNAs. But traces of the original chemical affinity might still be detectable. Whatever its precise origin, the genetic code represents a kind of molecular language, a symbolic system that maps between two completely different types of molecules, nucleic acids and proteins. The existence of this code is one of the most remarkable features of life. It's what allows information stored in DNA to be translated into the functional machinery of proteins. Without this translation system, DNA would be meaningless, just a sequence of bases with no phenotypic consequence. The last universal common ancestor. Our journey through the origin of DNA inevitably leads us to LUCA, the last universal common ancestor, the organism from which all life on Earth descends. LUCA represents the earliest point in evolution that we can reliably reconstruct using comparative genomics. By examining genes shared across all three domains of life, bacteria, archaea, and eukaryotes, scientists can infer what LUCA was like and what genes it possessed. Recent reconstructions of LUCA paint a picture of an organism that was already quite complex. A 2024 study published in the journal Nature, Ecology, and Evolution, analyzing phylogenetic data from thousands of modern genomes, concluded that LUCA lived approximately 4.2 billion years ago, surprisingly early, leaving only about 1 to 200 million years between the formation of habitable environments on Earth and the emergence of LUCA. LUCA appears to have been a prokaryote-like organism living in a hot, hydrothermal environment. It had a sophisticated genetic system, including DNA synthesis machinery, amino-acyl tRNA synthetases for the genetic code, and ribozymes for protein synthesis. It had cell membranes and the machinery for generating energy across those membranes. Remarkably, LUCA seems to have possessed elements of a CRISPR-Cas immune system, suggesting it was already engaged in an evolutionary arms race with viruses. The genome of LUCA likely contained around 2,600 protein-coding genes, comparable to modern prokaryotes. This is not the simple, primitive organism that early researchers imagined. LUCA was a full-fledged cellular organism with complex metabolism, likely an anaerobic acetogen that used hydrogen gas as its energy source and fixed carbon dioxide into organic molecules. But here's the key question for our story. Did LUCA have a DNA genome or an RNA genome? This question has been intensely debated. Some researchers argue that the differences in DNA replication machinery between bacteria and archaea suggest that LUCA had an RNA genome and that the transition to DNA occurred independently in these two domains. Others argue that LUCA already had DNA, based on the presence of certain DNA processing enzymes that appear to be universal. The truth is probably complex. LUCA might have had what some researchers call a mixed RNA-DNA genetic system, using both types of nucleic acids in different capacities. Reverse transcriptase, the enzyme that converts RNA into DNA, might have been present, allowing genetic information to flow in both directions. Such a system would have been messy by modern standards, but it might represent an intermediate stage in the transition from pure RNA to pure DNA genetics. What's clear is that by the time of LUCA, the modern system of DNA to RNA to protein was essentially in place. The genetic code was established. The fundamental machinery of transcription and translation existed. DNA had taken over as the primary repository of genetic information, though RNA retained crucial catalytic roles, especially in the ribosome. LUCA represents not the origin of life, but a later stage, a population of organisms that had already evolved for perhaps hundreds of millions of years. The true origin of life, the transition from chemistry to biology, happened earlier. The first genetic molecules, the first self-replicating systems, the first protocells, these all predated LUCA by an unknown span of time. LUCA is simply the most recent ancestor that left descendants in all three domains of life. Earlier forms of life might have existed and gone extinct, leaving no trace in modern organisms. The long period between the origin of life and LUCA allows for substantial evolution to have occurred. During this time, the RNA world could have flourished and then given way to the DNA protein world. Protocells could have evolved into true cells with sophisticated membranes and energy generation systems. Competition and natural selection would have winnowed countless failed experiments, preserving only the most successful designs. By the time we reach LUCA, we're looking at life that has already been extensively refined by evolution. The multiple origins question. One of the most profound questions in origin of life research is whether life arose only once or multiple times on Earth. Could there have been different origins of life, different experiments in creating self-replicating chemical systems with only one lineage, Luca's, surviving to the present day? The fact that all known life uses the same genetic code, the same handedness of amino acids and sugars, and the same basic biochemistry suggests a single origin. If life had arisen multiple times using different codes or different chemical systems, we might expect to see organisms today with fundamentally different biochemistry. The universality of DNA, RNA, and the 20 standard amino acids points to a single ancestral population. But this doesn't mean life only originated once. It's possible that life emerged multiple times, but that one lineage outcompeted all the others, driving them to extinction. Early in Earth's history, when different chemical systems were still competing, there might have been organisms using different genetic molecules, different codes, different metabolic strategies. Over time, the most efficient system, the one based on DNA, RNA, and proteins, might have come to dominate, either by being more successful or by absorbing useful innovations from other systems through lateral gene transfer. Some researchers have proposed that the universal features of modern biochemistry, the choice of certain amino acids, certain cofactors, certain metabolic pathways, reflect not absolute requirements, but historical contingencies. If life originated again today, even in a similar environment, it might develop a completely different biochemistry. The DNA-RNA protein system is just one solution to the problem of creating self-replicating, evolving systems. Other solutions might be possible, but we've never encountered them. This possibility has implications for the search for life elsewhere in the universe. If life on Earth reflects just one solution among many possibilities, then life on other planets might be radically different from anything we know. It might use different genetic molecules, different amino acids, different solvents instead of water. The challenge for astrobiology is to develop detection methods that don't assume Earth-like biochemistry, but can recognize any system that shows the hallmarks of life: replication, evolution, metabolism. On the other hand, if the DNA-RNA protein system represents something close to an optimal solution, then life elsewhere might converge on similar biochemistry. Just as eyes have evolved independently many times on Earth, perhaps genetic systems based on nucleic acids and proteins would evolve on other worlds. We don't yet know whether life's biochemistry is deeply contingent or strongly convergent. Discovering life elsewhere and finding out whether it resembles Earth life biochemically would help answer this question. The experimental frontiers. Today, researchers are making remarkable progress in understanding the origin of DNA through laboratory experiments that recreate possible prebiotic conditions. These experiments are testing whether the chemical steps leading from simple molecules to nucleotides to RNA to DNA can occur under plausible early Earth conditions. One frontier is the non-enzymatic synthesis of RNA. Researchers have shown that under certain conditions, nucleotides can spontaneously link together to form RNA chains without the help of enzymes. The key is providing the right environment, the right mineral surfaces, the right pH, the right concentration of salts. Temperature cycling, as might occur in a hot spring with day-night temperature swings, can drive the assembly of nucleotides and even promote the copying of RNA strands. Scientists have created ribozymes that can catalyze their own synthesis, demonstrating that self-replicating RNA systems are chemically possible. A recent study described a 45 nucleotide ribozyme that can catalyze the synthesis of both its complementary strand and copies of itself using simple trinucleotide building blocks. This shows that fairly short RNA molecules can achieve surprisingly complex catalytic functions, making the spontaneous emergence of self-replicating ribozymes more plausible than previously thought. The space of possible ribozymes is vast. Researchers have estimated that there could be more than 10 to the 39th power different RNA sequences capable of autocatalytic self-reproduction. This enormous number suggests that the origin of self-replicating RNA might not have required an improbably lucky accident, but could have been a relatively probable outcome of prebiotic chemistry, given the right conditions. Experiments have also demonstrated that RNA and DNA can coexist and be synthesized together from similar starting materials, challenging the strict version of the RNA World Hypothesis that assumes pure RNA came first. This mixed RNA-DNA world scenario might better reflect the messy reality of early chemistry, where many related molecules were present simultaneously, and evolution selected from this mixture rather than proceeding through neat, separate stages. Researchers are also working to create artificial cells in the laboratory, protocells that can encapsulate genetic material, undergo some form of metabolism, and potentially divide. These synthetic biology approaches test whether we can recreate the essential properties of life from the bottom up. If successful, they would demonstrate not only that life's origin was chemically possible, but would provide a working model of what early cells might have been like. One tantalizing goal is to create an artificial organism with a genome made of something other than DNA, perhaps an alternative genetic polymer that Earth life never evolved to use. This would prove that DNA is not the only possible genetic molecule, just the one that happened to succeed on Earth. Several laboratories have created XNA polymers, xenonucleic acids with different sugar backbones that can store genetic information and even evolve through directed evolution in the lab. These experiments show that the space of possible genetic molecules is larger than nature has explored. The philosophical implications. The origin of DNA raises profound questions that extend beyond chemistry and biology into philosophy. What is life? What distinguishes the living from the non-living? How do we draw the boundary between complex chemistry and simple biology? These questions don't have easy answers. Life seems to be a matter of degree rather than a sharp boundary. There's a continuum from simple chemical systems that can reproduce themselves imperfectly to more complex systems with metabolism and homeostasis, to systems with heredity and evolution, to cellular organisms with elaborate regulatory networks. Where along this continuum should we say that life begins? The study of viruses has sharpened these questions. Viruses contain genetic material and can reproduce, but they can only do so by hijacking the cellular machinery of their hosts. Are they alive? Most biologists say no because they lack metabolism and cannot reproduce independently. But viruses evolve, they have genomes, they respond to selection pressures, they occupy a gray area between chemistry and life. The RNA World Hypothesis suggests that the earliest life forms might have been even simpler than viruses, perhaps just self-replicating RNA molecules without any cellular structure. Were these molecules alive? They could reproduce and evolve, which are key characteristics of life. But they lacked the compartmentalization and metabolism we associate with cells. This suggests that life's emergence was gradual, a process rather than an event, with the living and non-living not separated by a sharp line but connected by a continuum. The origin of DNA also raises questions about inevitability and contingency in evolution. Was the emergence of DNA inevitable, given enough time and the right chemical conditions? Or was it a historical accident, one possible outcome among many? If we could rewind the tape of Earth's history and run it again, would DNA emerge a second time, or might evolution discover a completely different solution? These questions matter because they affect how we think about life elsewhere in the universe. If DNA's emergence was inevitable, then we might expect to find DNA-based life on other planets with similar conditions. If it was contingent, then alien life might be biochemically unrecognizable to us. The search for extraterrestrial life depends partly on answering these questions about how constrained or open-ended the origin of life is. The wonder of the code. As you drift toward sleep, consider the molecule that encodes your existence. Every cell in your body contains roughly 2 meters of DNA, coiled and packed into a nucleus just a few micrometers across. Stretched end to end, all the DNA in your body, say, would reach to the sun and back more than 60 times. This vast library of information, written in a 4-letter alphabet, contains the instructions for building and maintaining you. But this molecule, for all its importance to you personally, is part of a much larger story. The DNA in your cells is descended through an unbroken chain of replication and cell division from DNA that existed in organisms billions of years ago. Every time your cells divide and copy their DNA, they're performing a process that has been refined over eons of evolution, a process that links you to the deepest past of life on Earth. The origin of DNA represents one of the great transitions in the history of life, comparable to the origin of cells, the origin of photosynthesis, or the origin of multicellularity. It was the innovation that allowed life to store information reliably enough to build complex organisms, to preserve genetic instructions across billions of cell divisions, to create the biological diversity we see around us. Yet DNA did not spring into existence fully formed. It emerged from simpler precursors, from RNA molecules, and possibly from even earlier genetic systems we can only guess at. It arose from prebiotic chemistry, from the churning of molecules in an early Earth environment we're still trying to understand. The path from simple organic molecules to the elegant double helix was long and circuitous, filled with dead ends and false starts that left no trace. We stand now at a remarkable moment in human knowledge. For the first time in history, we can ask serious scientific questions about life's origin and expect to find answers. We can perform experiments that recreate early Earth conditions and watch complex chemistry unfold. We can analyze the genomes of thousands of organisms and reconstruct the properties of ancient ancestors. We can search for life on other worlds and consider whether the biochemistry we find will resemble our own. The mystery of DNA's origin is not fully solved. We still don't know exactly where and how the first nucleotides formed, how they assembled into functional polymers, how RNA gave way to DNA, how the genetic code was established. These questions remain at the frontier of scientific research, drawing together chemists, biologists, geologists, and astronomers in a collaborative effort to understand our deepest origins. But we know vastly more than we did even a few decades ago. We know that the building blocks of life can form naturally under plausible early Earth conditions. We know that RNA can act as both genetic material and catalyst, solving the chicken and egg problem of life's origin. We know that protocells can self-assemble from simple molecules. We know that evolution can create complexity from simplicity given enough time and the right selective pressures. We know that DNA is not eternal and unchanging, but is itself a product of evolution, a refined solution to the problem of information storage that emerged after billions of years of earlier evolution. We know that the code written in DNA is not arbitrary, but shows signs of optimization, of being shaped by selection to minimize errors and maximize efficiency. And we know that this code connects all life on Earth. The same genetic language is used by bacteria in deep-sea vents, by trees in tropical rainforests, by eagles soaring over mountains, by whales in the ocean depths, by you as you lie here preparing for sleep. This universality speaks to our common ancestry, to the fact that all life on Earth shares a single origin, a single moment billions of years ago when chemistry became biology and the first genetic code was written. The continuing journey. The story of DNA's origin is not just a story about the past. It's a story about the present and future as well. Understanding how DNA emerged helps us understand how life works today, why it has the particular biochemistry it does, what constraints and possibilities exist for living systems. This knowledge has practical applications. Synthetic biologists are using principles learned from studying early evolution to design new genetic systems, new metabolic pathways, new organisms with capabilities that natural evolution never produced. By understanding how DNA works and where it came from, we gain the power to modify it, to edit genomes with precision, to write new genetic code. This power brings responsibilities. As we learn to manipulate the code of life, we must grapple with ethical questions about how this technology should be used. But these are good problems to have. The problems that come from understanding rather than ignorance, from capability rather than helplessness. The search for life elsewhere in the universe is informed by our understanding of DNA's origin. As we explore Mars, examining rocks for signs of ancient life, we're guided by knowledge of what chemical signatures might indicate biological activity. As we study the icy moons of Jupiter and Saturn, planning missions to search for life in their subsurface oceans, we consider what kinds of organic chemistry might occur in those alien environments. As we analyze the atmospheres of exoplanets orbiting distant stars, looking for biosignatures, we use our understanding of Earth life's biochemistry to guess what we might detect. The discovery of life elsewhere would be profoundly illuminating for understanding DNA's origin. If we found organisms using DNA and the same genetic code, it would suggest that these solutions are somehow optimal or inevitable. If we found organisms using completely different genetic molecules, it would show that life's biochemistry is more contingent than we thought. Either way, we would learn something fundamental about the nature of life. But even without finding life elsewhere, the study of DNA's origin teaches us something profound about our place in the universe. It shows us that we are not separate from nature, but are part of an unbroken chain of chemical and biological evolution stretching back to the earliest days of Earth. The atoms in our bodies were forged in stars. The molecules in our cells arose from reactions between simple chemicals on the young Earth. The genetic code that defines us emerged through billions of years of evolution. We are, in a sense, the universe's way of understanding itself. The same physical and chemical laws that created stars and planets also created life and mind. The same carbon atoms that once floated in interstellar space now form the DNA in your cells, encoding your memories, your thoughts, your capacity for wonder. You are made of stardust organized by evolution into a form capable of contemplating its own origins. Closing thoughts. As sleep approaches, let your mind rest on this. You exist because of a molecular code that emerged from chemistry billions of years ago on a planet that was nothing like the world you know. That code has been copied with small variations through countless generations, through mass extinctions and evolutionary radiations, through the rise of bacteria and the emergence of complex cells, through the evolution of plants and animals, through the lineage that led to humans and ultimately to you. Every organism that has ever lived carried some version of this code. Every tree that has grown, every bird that has flown, every fish that has swum, every bacterium that has ever divided, all used the same fundamental system of genetic information, the same DNA molecule with its 4-letter alphabet, the same translation system converting genetic sequences into proteins. This code is both ancient and new. Ancient because its basic form was established billions of years ago, before complex life existed. before oxygen filled the atmosphere, before continents had formed. New because every time a cell divides, the code is freshly copied, and errors in that copying introduce variation that drives evolution forward. You are a temporary custodian of this code. The DNA you carry was given to you by your parents, who received it from their parents, stretching back through your ancestors to the beginning of the human lineage, back through earlier primates, back through the first mammals, back through the first vertebrates, back through the first animals, back through the first multicellular organisms, back through the first eukaryotes, back through Luca, back through the RNA world, back to the first self-replicating molecules that could be called alive. And you will pass this code forward, whether through children who inherit your genes, or through the impact you have on the world, or simply through being part of the great web of life that shares this planet. The code continues, copying itself, varying, evolving, creating new forms while preserving the essential pattern. The origin of DNA is ultimately a story about information, how information can emerge from chemistry, how it can be stored and copied, how it can guide the construction of complex structures, how it can evolve and adapt. Information is perhaps the most fundamental aspect of life. Without information that can be reliably transmitted across generations, there can be no heredity, no evolution, no biology. DNA solved the problem of information storage so well that it has persisted for billions of years. It may persist for billions more as long as life continues on Earth. When the sun eventually expands and makes Earth uninhabitable, perhaps our descendants will carry DNA to other worlds, spreading the code that originated here across the galaxy. Or perhaps by then, evolution will have found something even better, a new solution to the old problem of how to store and transmit information reliably and enough to support complex evolving life. But for now, DNA is the code of life. Its origin, shrouded in the mists of deep time, represents one of the great mysteries that science is slowly, patiently unraveling. Each experiment brings us closer to understanding how chemistry became biology. Each fossil gives us another clue about early life. Each genome sequenced helps us reconstruct the path evolution has taken. We live in an age when we can read the code that defines us, when we can trace our ancestry back through billions of years, when we can ask questions about the origin of life and expect to find answers. This is a remarkable privilege. For most of human history, our origins were a complete mystery, explainable only through mythology. Now we can do better. We can use the tools of science to illuminate our past. The story is not finished. New discoveries await. Perhaps researchers will soon create self-replicating RNA in the laboratory, demonstrating conclusively that the RNA world is chemically possible. Perhaps we'll find fossilized evidence of early life that clarifies what the first organisms were like. Perhaps we'll detect life on another world and discover whether it uses DNA or something completely different. Perhaps we'll finally pinpoint exactly where and how life originated on Earth. These discoveries lie ahead, waiting for future scientists to uncover, but already we know enough to be awed. The molecule coiled in the nucleus of every cellular cell in your body has a history stretching back billions of years. It emerged from simpler precursors through processes we're still working to understand. It has been refined by evolution into an exquisitely efficient system for storing and transmitting information, and it connects you to every living thing that has ever existed on this planet. As you sink into sleep, consider this inheritance. You are made of matter that has been part of the universe since the beginning, arranged by evolution into patterns of astonishing complexity. The code that defines you was written over billions of years through the patient work of variation and selection. You are a temporary expression of that code, a brief moment in its long journey through time. Tomorrow you will wake, and your cells will continue copying their DNA, maintaining the code, passing it forward. But tonight, rest in the knowledge that you are part of something vast and ancient, a story that began when the first molecules learned to replicate themselves, and that continues in you at this very moment. If you found this exploration of DNA's origin thought-provoking and enlightening, please consider subscribing to DreamLab Knowledge. Hit that like button to help others discover this journey through the deepest mysteries of biology and chemistry. Share this with anyone fascinated by the origin of life, by the molecular basis of heredity, by the question of how chemistry became biology and information emerged from matter. Your engagement helps us continue producing content that tackles the profound questions about our origins, that brings together insights from multiple scientific disciplines, that values both what we know and what remains to be discovered. Together we can build a community that embraces the wonder of scientific discovery, that sees in the double helix not just a molecule but a connection to the deepest past, that finds meaning in understanding our place in the vast sweep of cosmic and biological history. Thank you for joining this journey into the origin of DNA, the code that connects all life on Earth. And until next time, keep questioning, keep wondering, and keep marveling at the extraordinary fact that matter can organize itself into forms capable of understanding their own origins.